I'm Dr. Greg Nye, and welcome to another exciting edition of the Deep Dive with me. And today I'm going to be going through a paper that I was co-author on with Dr. Stephanie Seneff of MIT. This is one of the first really, well, not the first, but a significant controversial paper that we published. This was on the vaccination, the mRNA vaccinations. And just to give, in fact, I'm going to share my screen so that you can kind of follow along with this. Okay, hopefully it's going to show up here. There it is. So the paper is called Worse Than the Disease, Reviewing Some Possible Unintended Consequences of the mRNA Vaccines Against COVID-19. So the context is that in 2020, going through that year, Stephanie and I had been working on a different project, and then it was all this talk about the mRNA vaccinations was sort of making its way into the public. And we started looking into it and we're like, oh crap, we got a problem. I'll kind of put this in the context of this phrase that is commonly heard, or, signs that people put in their yard, something like follow the science or trust the science. It's a completely bogus phrase. There's no such thing as the science. It is a process done by people who all have various interests, and there are an enormous number of factors that come into play regarding what even gets published as science. There is no such thing as the science. And if anyone in a position of authority within the world of health or even other areas of science, if they ever say to follow the science or trust the science, they are manipulating you. Look the other way. Something ain't right with anybody who advocates the position that there is such a thing as the science that needs to be trusted. Okay. In that context, Stephanie and I started looking at what had been published, what was already known about the various components that were going into these mRNA vaccinations. This is in 2020. Keeping in mind, the vaccinations came onto the market in really December of 2020. They started being introduced to elderly and healthcare workers. And it wasn't until spring of 2021 that they were then kind of released for public use. And vaccination, by the way, we'll put that in quotes because maybe I'll talk about that another time. We started looking into it, like what was available within the scientific literature that would suggest that maybe there's going to be a problem here. So over the course of 2020, kind of did that homework. Oh my gosh, there we go. Oh, first of all, let's just put this in context. There have been those who defended the vaccinations by saying, oh no, these have been in development for a decade. Like mRNA vaccination is old. This is just a new application. That was not true. That was an absolute distortion of what was happening. Just to give an idea of the timeline, I got these documents, this is out of the paper, but here's a graph that explains the typical time it takes from the idea of something you want to develop a vaccination against and how long it's going to be until that is actually through the testing and ready to be put on the market. So the average, there are three kinds of vaccinations, which I give you here am I still sharing the screen? I hope. They are simple, complex, and unprecedented, which is literally that's the category of the vaccination. Simple is just using what's been done in the past. Complex is a modification of what's been done in the past. Unprecedented means there is no history that you can base the new one on. You're coming up with an entirely new vaccination. So, let me get this out of the way. So, average cost of development, this is giving you the cost, the time per phase in years. So, pay close attention here. The average time per phase to test an unprecedented vaccination. You can see for both complex and unprecedented, you're looking at over 12 years. 12 years to get these things tested so that they're ready to get to the market. And the probability that that any given one is going to make it through all phases of testing and ultimately be approved for the market for an unprecedented vaccination, there's a 5% probability that it's going to make it through all that over the course of 12 years. This vaccination developed by Moderna, a company that had never marketed a single product before this one. It went from what was called testing to market in an insanely short time. We're talking like 6 months, not over 12 years, 6 months. And the first one that they tried, I'll be damned if they didn't get it to market, even though there's only a 5% chance historically for any given attempted vaccination to make it to market. This one made it to market by a company that's never done it before. Gosh, maybe there could be problems in that. Okay, what Stephanie and I did was to just look at the information that was available about how these lipid nanoparticles, about all the different components of the vaccine, both the spike protein and how it had been modified, and also the carrier for it. So there were these cationic, positively charged lipid nanoparticles that were the carrier for the mRNA. Keeping in mind, positively charged lipids do not exist in our bodies. We have negatively charged lipids. They're not positively charged. We don't have enzymes for breaking down positively charged lipids, but these were made in positively charged lipids, and they contained something called PEG, polyethylene glycol, as well as some other components, cholesterol and some other things. Okay, so we need to look to see, are there potential for those components to cause consequences that we don't want to have? With the spike protein itself, there are several genetic manipulations. So the thing that people were getting via injection, mRNA, to code for a spike protein, that protein that was being coded was not like the one that was in circulation, that if somebody got exposed to by natural infection. They were not the same. There were many genetic manipulations that were done so that the new protein that got made was different than the one in nature. And other manipulations were done to prevent this mRNA from getting broken down before it could produce any proteins, because that's a big problem. That was one of the big hurdles they had to overcome, was to prevent the degradation of that mRNA. They had to make it live a long time. So one of the ways they did that was by replacing, mRNA is made of, it's like beads on a string, and what they could do is replace some of those beads, called uridine, with some other beads called N-methyl pseudouridine. When they made that substitution, when mRNA has lots of uridine in it, it's a danger signal to the body that, oh, we gotta degrade that. That is not supposed to be here. But when you replace it with N-methyl pseudouridine, the body doesn't pay so much attention to it. It allows that mRNA to hang out in the body, which is what they intended for the mRNA to do, so that it could keep producing spike protein. But now, with that one substitution, you've just subverted one of the ways that the body protects against the accumulation of RNA, which is a danger signal for a cell. Prolines at these two spots. So proline is another amino acid that when you build the spike, you got different amino acids there, and proline is one of them. They spliced in two prolines. The effect of that splicing was to allow the spike protein to get lodged in the cell membrane and not convert to its post-fusion. It's called post-fusion stabilization. So it stabilizes it in the cell membrane so that it doesn't go away. Normally, with natural exposure, those prolines aren't there. It allows the spike protein to fold. When you don't allow the folding, it prevents it from getting cleared from the cell membrane. So once again, they're creating it in a way that it's going to persist in the body. maybe that's a problem. Something called CG enrichment. This is replacing, before we talked about uridine getting replaced. Now we're talking about a few other bases. Doesn't matter, but so just a series of bases, 4 bases in RNA, you replace some with something else. And in doing so, you increase the lifespan or the expression of the RNA by at least 100 times. So not only are they building the RNA so that it doesn't get degraded, but the RNA that gets in there is now expressing itself at least 100 times more aggressively than normal RNA does. So it's built so that it doesn't degrade and it doesn't turn off, and it is in this overdrive in terms of how much it's going to express. And in this case, that means generate spike protein. Where's the off brake, you might ask? Where's the stop button? Doesn't exist. It doesn't exist. Then we have the biodistribution issue. Again, these are just things that Stephanie and I were looking into to say, could any or all of these lead to unintended consequences? If the real goal was just to prevent people from getting sick by getting exposed to natural coronavirus, could any of these things that they're doing to this sequence of RNA cause problems? So one of the things that was known back then, that this RNA distributes to the spleen, the lymph nodes, the liver, the brain, the testes. And since then, we have now learned that it is essentially system-wide. It goes to the brain, it goes to the placenta, it goes to the bone marrow. It is widely distributed, even though the health officials were saying, oh no, it's going to stay in the arm. That's what was said about this. But it was known, if they had followed the science, they could have seen that this was going to be a problem. We were just accessing what was publicly available within the scientific literature. So then the issue of GXXXG motifs. So within amino acid sequencing, there's a glycine and then 3, any amino acid, XXX, and then a glycine. So these are they give a cell a strong tendency for making prions. Prions, maybe you've heard, associated with mad cow disease or Creutzfeldt-Jakob disease in humans, or Parkinson's and Alzheimer's and even MS and other neurological conditions, degenerative bad things. Built into this spike protein that's being used to inject into people, built into it are 5 of these GXXXG motifs, they're called. Times that happens. So the amyloid prion protein, the one that's associated with Alzheimer's disease, has 4 of those sequences, making it very prone to becoming a prion protein. Spike, this thing has 5 of them. It has more than the one associated with Alzheimer's. So that's something we wanted to pay attention to. Another issue has to do within the manufacturing process. This was in documents that Pfizer presented to the FDA to show that. So if you are generating sequences of RNA and you're relying on bacteria to spit these things out, but maybe it doesn't generate the entire sequence. Maybe it has fractions of sequences, because at some point you got to stop the growth. You got to add things that then shut off the production. And so maybe you've got some full-length RNA, but then you have these other fragments of RNA, because different cells are at different stages of generating the RNA. So what happens with those fragments of RNA? Well, as RNA fragments, they do what they're intended to do. They generate protein sequences. But these now are not going to be functional proteins. They're not going to be the spike protein that the full RNA makes. Now we've got fragments of RNA. What are they going to make? Well, we don't know. Pfizer didn't know. They presented that to the FDA. The FDA said, we need you to tell us, are these fragments going to produce any kinds of proteins that are detrimental to health? They put that assignment back on Pfizer. They never did it. They did not do that. They never followed through with determining how much of these fragments of RNA are then going to generate proteins, or what might be the consequences biologically of having these sort of like mutant proteins hanging around. We don't know. Let's see, what else? Sequence homology. So, another thing that we looked at was okay, so how similar is this spike protein to other proteins in the body? Because if I generate an antibody against spike, which is what they're trying to make us do, if the spike looks like collagen, which is the most abundant protein in our body, if any aspect of this spike protein looks like this collagen protein, then this antibody could also bind to this collagen. So we just looked at what is the literature, how many different proteins in the body have homology with this spike protein that is getting injected. And in fact, there were studies out there that showed a very large number of very important proteins have homology with the spike. So if we're going to generate antibodies to these, we could very well start generating antibodies to these others that they have homology with. And some of them, I just mentioned the TPO protein associated with the thyroid, PTG is associated with celiac disease, collagen is essentially all of our connective tissue is built of collagen. The autoimmune issue is big, and we felt it was pretty important to give some attention to that. And of course, as it turns out, yeah, there is this significantly increased risk of several kinds of autoimmune diseases that are now manifesting following the vaccination campaign. Many are trying to attribute that to natural infection, that, oh, it's just COVID, and COVID is causing people to have more autoimmunity. I think that there are really solid scientific reasons to doubt that. That's not to say that COVID couldn't promote any of these autoimmune problems, but the risk of it would be much less from natural immunity. A final thing that we put on here that we were looking into, and we have a section of the paper about, has to do with shedding, is this idea that, and first of all, this is a normal process. When we have infections, viral infections, cells will take up the virus, and then in order to like send out a danger signal to other cells around the body that you got to get ready for this, they'll package the virus in what's called an exosome, along with some other little bits of RNA that code for some immune activators, because it's like, okay, here's the package. We're going to present you with the intruder that you need to watch out for, and here's the code to help you make the immune molecules to combat it. It's brilliant. Pretty brilliant. So cells do this as a natural thing, and those exosomes can be released into circulation so that they get distributed around the body so that other cells are told about what's about to happen, and they can come out through the breath and the skin. They can be secreted out. This is not some conspiracy, whatever. This was known when Pfizer submitted their guidelines when they were testing the vaccination before it got released. They had very specific guidelines about after people had received one of these vaccinations, they were not supposed to be in physical contact with their partner or spouse. They had to report if they had skin contact, because they knew that there was this shedding problem that could be coming up. Now, of course, there have been some studies done where they compile evidence. I can tell you from my own clinical experience, it is without question that there are some people who are absolutely developing symptoms. These are unshot people who get in a closed environment, like a family reunion kind of a thing, with lots of other people who got the shot, and they develop dramatic symptoms within a very short time after that exposure, symptoms that they had never dealt with before. So I feel confident from my own observation that shedding is a real thing, leading to significant health problems for at least some people. I'm not claiming it affects all or even most, but for the people it affects, it's a big deal. Let's see, do I have any more slides? Oh, yeah. So then essentially, we had all of this stuff that we could see documented about either the way it was built or what it was expected to do in the body, and all the ways that they manipulated it to keep it persisting in the body. And in fact, protein the generation of spike protein has now been documented to persist for, I believe, 3 years is as long as they've got it documented now. And I can tell you there are people who got a vaccination long previously who, when you test them, some of them again, they are still generating a very high level of antibodies against spike protein. Now, that is a persistent spike protein. There's one thing I didn't I guess, did we put in here one last piece in our paper that we actually speculated about was DNA integration. And we were assured many, many times by those public officials that, oh no, this can't integrate into DNA, our own cell DNA, because this is RNA, and RNA can't integrate into DNA. Well, that's simply untrue. That is very explicitly untrue. And they knew it. They had to know it. It is well established that cells have mechanisms for what's called reverse transcribing RNA into DNA, and then that DNA can incorporate into cells. This is not some obscure realm of science. This is very well known. And so in our paper, we kind of go through what made sense to us as the risk that this injected RNA could get reverse transcribed and then incorporated into host DNA, meaning integrated into our own DNA, so that our DNA is now capable of producing spike proteins for the rest of our life because it's built into the DNA. So we described the mechanisms for all of that. Then, the next piece was to get into, all right, basically, here's how it's built. Now, what are the implications? Like, what do we see happening? So we talked about anaphylaxis and antibody-dependent enhancement, which is to say that people who gotten the shot, because of the type of antibodies that it generates, it could make them even more susceptible to more severe illness if they get an exposure to this. Multisystem inflammatory syndrome is mostly diagnosed in children, but essentially we're saying that this, because of the immune activation that would come with the lipid nanoparticles, which again can persist very long time. They embed in the tissue. We don't have enzymes to break them down. They persist and they act as an inflammatory agent. They were intended to. That's one of the reasons they were used, is to provoke the immune system. But the problem is there's no off switch. They didn't build in an off switch. This inflammatory stuff, the autoimmunity we talked about, the pathogenic priming, which are so in fact right here, spike antibodies reacted to 18 out of 55, I think I have my numbers wrong, of the endogenous, meaning internal proteins that it was tested against. 18 different proteins that we naturally produce. If we make an antibody against the spike, we run a real risk of generating antibodies to any one of those 18 proteins that are natural to our own body. That is the definition of an autoimmune disease. One of the areas of the paper that we discussed quite a bit was the potential that the spike protein, because of the type of inflammation that it creates and the various pathways that it either shuts down or activates, we're going to see clotting problems. That there would be thrombocytopenia, which is a depletion of platelets, which are, of course, needed for clotting, or other of the clotting mechanism that would either hyperactivate or be impaired. So we had predicted that. And in fact, clotting has become maybe the most significant health consequence to follow from getting one of these shots or getting multiple of the shots. It's after the third one that the big problems really tend to set in. But for many people, it's even after just one. But the clotting leading now to heart attacks and strokes, leading to pulmonary embolisms, many manifestations now showing up of this excess of clotting that is being provoked by these shots. And again, this was all predictable. This wasn't like, oh my gosh, how did this happen? It was all there in the science. Just had to follow it. Latent virus activation. This is something also a section in the paper that we had said, oh, well, given what we know about how this spike protein is impacting the immune system, we would expect that viruses like herpes virus, and then I don't know if we mentioned in that paper, but we did mention in others about like Epstein-Barr virus, hepatitis C virus, some other viruses that can be just quiet. They are there, but they are not actively dividing. With these shots, we might wake them up and create a new active disease. So with herpes virus, we're talking about shingles and cold sores, which, actually, there's now been this correlation of a rise in the number of shingles cases that has come about since, oh my gosh, about 2021 when the shots were introduced. So that was another problem we talked about. We have a whole section on the direct toxicity of the spike protein, and the ways that it damages the artery wall. And it's just a toxic protein. We created a shot and injected people with the machinery that causes our own cells to make a toxic protein. It's madness. We, of course, as I mentioned, we predicted this rise would be expected in prion-associated diseases, MS, Parkinson's, Alzheimer's, and other even more serious dementia. And in fact, that has been happening. And I think currently, when they look at increases in diseases, when they do studies now, they're looking at like from 2010 up until 2020 or something. And they say, oh yeah, we see a rise. The problem is we got to get a little further out before they start looking at like 2020 to 2025 or 28, because that will cover the COVID time. And I feel really confident that there's going to be a dramatic increase in the diagnoses of these prion-associated conditions because we're injecting a protein that can become a prion protein. We already have seen dramatic rise in heart disease and strokes, and also dramatic rise in the incidence of new cancers being diagnosed. And in fact, at some other point, I'll be talking about a couple of papers that we published predicting the rise in cancers because of just many of the ways that this spike protein is interfering with cancer surveillance. Anyway, I'll get to that. Novel emerging strains due to getting the shot. So, and this, I won't go into this too much, but basically, if you inject a spike protein, or you inject the genetic sequence to produce a spike protein, if you then keep generating antibodies to that spike protein, well, it just mutates itself so that it's building a kind of protein that is not exactly like the one that the antibody can get. So you induce new strains of this by injecting people. And this is documented. We have it in the paper. This really did happen. And again, it's not because we got some freaking magic ball that we're looking into to predict things. This was in the literature. This should have been making headlines, but that's how it goes. And then finally, we had predicted the DNA incorporation. And now, I won't get into it all, but yes, that looks very much to be the case. And in fact, this has become quite a big deal and is sort of, they're trying to make it a contentious idea that it's really happening, but it's not contentious at all. The evidence is really clear that not only does it incorporate into DNA, but in ways that we could not have imagined back then. It was designed to be incorporated into the DNA. So that is maybe for another podcast. But so, all right, this ended up longer than I thought. Maybe I'll try to squeeze the next ones down. But this paper was a big one that we put together and essentially did all the research on it before the vaccinations were even introduced. This could have been publicly discussed so that people could hear some of these potential harms, and then they can hear like maybe what some benefits are if they get the shot, so that they can make informed consent. But that was done away with. As a last footnote, one of the ways academic censorship works is to just keep papers from getting published in a journal that is indexed in PubMed, which anybody who's doing medical research, they search PubMed. All those journals are indexed on PubMed. And if you want research not to get indexed on PubMed so that nobody sees it, you don't allow it to be published in a journal that is indexed there. So we wrote this paper, we submitted it to several journals. We feel like the science that we packed into the paper couldn't really refute it. But of course, journal after journal rejected it, saying, it's not really the kind of thing that we publish. Even though it was exactly on the topic and matched other kinds of things that they published, they said no. Ultimately, we got it published in the Journal of in an International Journal of Vaccine Theory, Practice, and Research, not indexed on PubMed. But fortunately, world of the internet, even though it's not, once we got it published, we were able to distribute it on social media, and we even got it. Tucker Carlson actually featured this paper in one of the segments when he was on Fox News. So it did get some attention, but the censoring of it, the keeping this kind of information, the science on the vaccinations and the potential consequences, they didn't want the public to see it. So this is a topic that I'll be talking a lot about over the course of the, so won't go into it more here, but I appreciate you're following along. If you have any questions or comments about the kind of stuff that I brought up related to this paper, feel free to put that in the comments, and please do the subscribe and distribute this to whoever you think might be interested. Right. Thanks for being here today, and I will be back again before long.